Okay, hello, I'm running a bit late, sorry everybody. Good afternoon. Here we go, right. I'm starting to wonder if the name of this is becoming slightly inappropriate as lockdown learning. I'm wearing a shirt today for the first time in about 4 months, as it's starting to look less and less like lockdown, at least around here anyway. So, how is everybody? Good, I hope. It's Wednesday afternoon again, and for my small ones, it's their last week of term, so summer holidays are looming, but we are going to do some lockdown learning today. So, a little bit of racking my brains to think about what we're going to do this week, and I'm going to wing it slightly, but we are going to do some bioinformatics. So we've done a few weeks now of kind of technology tutorials, really learning about some particular technologies relevant to bioinformatics, but not necessarily not directly bioinformatics, as in analysing biological data. And so I think what I'd like to do this week is to think a little bit about some actual bioinformatics again for the first time in a while. So I'm going to talk about genome assembly, particularly prokaryotic genome assembly, but everything kind of applies to larger genomes as well. So we'll see how much we can get through, but I think we'll take a look at some of the principles behind genome assembly and how a lot of the major algorithms work, and then we will look at assembling. So we've got some E. coli reads that we've downloaded, so we'll use those E. coli reads, which we've aligned to the E. coli genome in lessons past, and we'll try assembling them instead, which is a slightly different bioinformatics procedure. So I'm also trying something a little bit new, which is a new whiteboard, but it's already not working. So I'm just refreshing myself here while I'm hiding from view. Let's see if this works. Okay, that looks like it might be working, sort of. So, okay, here we go. Right, we have talked previously about aligning, so the aligning of reads to reference genomes, which we have done using Bowtie 2. One of the other things that I want to talk about at some point is different aligners, because I realised that I introduced Bowtie 2, but I haven't really talked about other alignment strategies, and particularly for RNA-Seq, other alignment strategies are required. So we might talk about that in the future. But alignment is not a straightforward problem, but it is perhaps different to assembly, which is what we're going to think about today. So with alignment, you have some sort of reference genome. So you have an idea of what your sequence should look like, and then the process of alignment takes your reads and finds where they belong on the reference genome. Okay, so you align them. Align your reads to your reference, and then you come up with this alignment, which is usually in a BAM file, as we've discussed, and that is a kind of well-trodden path, and we've seen that kind of process of alignment, and there are good alignment strategies that exist for doing that. So we talked about those, and we talked about the data transformations that are required. Assembly is a slightly different class of problem. So with assembly, you don't have that reference genome. So the reference genome, the kind of idea of the reference genome that you have in alignment, that's not there. It's not present. So instead you just have these reads disconnected from any idea of where they might belong on your genome. And so in order to figure that out, in order to figure out how the placement of the genes in the context of the genome, you instead of aligning them to this non-existent reference genome, you instead have to compare them to each other. So you have to think about the relationship between not between your reads and the reference genome, but between your reads and each other. Okay, so without the presence of the reference genome, with that object missing, we have to think about the relationship of the reads to each other. And that is the essence of de novo genome assembly. So we're not thinking about reference-based assembly. We're considering that we don't have a reference, and you can do this with E. coli as much as you can with anything else. You just ignore the fact that there's a reference genome out there. So there are lots of different approaches for doing this. The one which we're going to look at today, and the one which is which these days is used most frequently, or the algorithmic approach which is used most frequently uses a structure which is generated from your reads called a De Bruijn graph. I can't spell it. De Bruijn. That's it. I think it's got J in it. Graph. So a De Bruijn graph is a structure that's generated from the reads, which allows you to figure out the overlaps between the reads in an efficient way. And the way that it's constructed is very specific. So if we take an example, and I think what I might do actually is, rather than drawing this all out, I might pull up some slides, because I do have this. I do lectures on this, so I might just grab some lecture slides and see if that's a slightly better way of talking about this. Okay, so here we go. Here's an example of this is a very simple assembly problem, and we're going to use this just as an example of how to build a De Bruijn graph from some sequence data, and how we then navigate that graph to assemble genomes. So in this case, we have a very simple genome of just 10 bases, and we have two 7-base reads, which we can assemble via a single perfect overlap to assemble that genome. So this is obviously a very trivial problem in terms of genome assembly. But the key thing to the way that a De Bruijn graph works is this parameter, which is known as K. Okay, so the parameter K is how we define how the graph gets built. So this is the example using a value of K of 4, and what K defines is the size of fragments that you're going to make from your reads. So you're going to break your reads up into fragments of length K, overlapping fragments of length K. So here we have taken our two sequences, sequence 1 and sequence 2, and we've broken them up using a value of K of 4. So we've broken them up into overlapping formers. So we've got these 4 formers from sequence 1, on the left, and these four formers from sequence 2 on the right. And from these fragments, so from these k-mers, we're now going to build a De Bruijn graph. And the way that we build that graph is we make some nodes. So here are nodes, and these are all sequences of all the sequences from our k-mers of length k minus 1. So we've taken our k-mers on the left here, and we've built them into nodes in a graph with all of the sequences of k minus 1. So ATC, TCG, the two nodes that you get from this first k-mer, and then TCG is repeated, and then you get GCT here as well. So you just get all of these nodes that we build from our k-mers, and then we define the edges between these nodes, and the edges are defined by nodes with an overlap of k minus 2. So any nodes which share their, in this case, their last two and their first two letters. So ATC and TCG share TC. That's a sequence of length k minus 2. So we draw an edge between those two nodes, and so on. And so this is the graph structure that we build by following those rules for this particular these particular two reads using a value of k of 4. And what you see is it's then hard to define a single path through this graph, because you've got several places where you can transition between the top and the bottom. So you can get from the ATC here to the AAT here by several different paths. Okay, so you can go along here and then down and then across and then along and then down and then across and then along, all the way along to the end and then down and then across. But none of those is there's not a single path through this graph. There's not a single path which takes in every edge in this graph, which means that you end up having little cycles and things like that, and you can't build a perfect representation of your genome. Or rather, you can build a perfect representation of your genome, but there are also other assemblies which you can build from this graph. So this tells us that the value of using a value of 4 for k is not ideal for this particular genome and these individual reads. So let's try a different value of k. This is a k of 5. So here are the k-mers, here are the nodes in our De Bruijn graph, and here are the edges. Still not perfect. We've got these two transition points between our two reads here, and so we still can't define a single path through this graph. So k of 5 is still too big, still too small. Sorry, a k of 6 on the other hand allows us to build a graph. So we're building the graph by the same means, and this time with our graph we can define a single path through this graph. So there is just one route through this graph from one end to the other, and by following that path through the graph and adding the contents of the nodes to each other as we go, we can rebuild our genome sequence up at the top here. So this allows us to perfectly assemble our genome sequence. So by taking these two reads of length 7, breaking them up into k-mers of 6, of which there's only 2 in each read, and then building our De Bruijn graph from those sequences, we can perfectly reassemble our reference genome. If, on the other hand, we go to a k of 7, well, that's the same length as the reads, and if we could assemble our genome from that, well, then why are we bothering to build a De Bruijn graph? Why are we bothering to kind of take this approach at all? So that's it's not necessarily the best, most elegant example, but it kind of works to show how the choice of k is kind of the essential parameter optimization for assembling a reference genome. For assembling a genome in a de novo way. Obviously, in a real experiment, the value of k is not quite so simply defined. So in a real experiment, you've got many more than just 2 reads. So you have lots and lots of reads, usually hundreds of thousands or millions. The overlaps between each of those reads is not going to be the same. So even with sort of perfectly random genome fragmentation, your overlaps between different reads is going to be subtly different all of the time. So there isn't really a single value of K that is correct for a real genome assembly. But nevertheless, there are ways and means of estimating the kind of best fit parameter for the particular data that you're working with. So we're going to work with our E. coli reads that we looked at earlier on in lockdown learning. So I'm going to bring up my virtual machine. So I'm just starting VirtualBox. And I'm going to bring up my virtual machine. And let's take a look at those reads and figure out if we can estimate a value for K for assembling this genome. So my VM's just booting up, and we'll just track down those reads. So this is taking rather longer than I expected it to. There we go. Slowly but surely booting up. Here we are. Okay, so let's start our terminal. And let's cd into the place where these reads are, which is in share week. I'm going to guess 10 or 11. I'm not sure which. There we go. It's in 10. And we do actually have the FastQC here, which is important because we need to know how many reads there are and how long they are. So let's figure that out by opening this multi-QC report. So I'm going to open open the file manager, go into share week 10 and open the multi-QC report, which I can't see. Where is it? There. So we'll open that up. That should open in our browser in theory. Yep, there we go. So this will tell us how many sequences we have. So the answer is 1.4 million sequences. In paired end, so it's paired end reads. We've got 1.4 million, roughly speaking, sequences. I wonder if we can get an exact number. Let's have a look at the table rather than the plot. So actually the data for this is in the multi-QC data folder, and we can have a look at multi-QC general stats, and that should tell us how many. There you go, 137. Sorry, 1,379,315 reads. So that's an exact number. We've also got the average length, sequence length in here, which is quite useful. So that is the 1, 2, 3, fourth column. So the fourth column is the average length, which is 208 bases. Okay, so those are useful statistics for us for estimating our value of k. So our bases are on average around about 208 bases long, and there's 1,379,315 of them. It would also be useful to know exactly how long the E. coli reference genome is. That's a useful thing to know as well. I know it's about 4.5 megabases. We can figure that out from the FASTA. So if we do everything from the second line on in the reference FASTA, so that excludes the headline, and then we're going to count the number of characters. That will tell us how many characters are in that reference genome. So there you go, 4,699,673 bases in the reference genome. So the next job then is to try and figure out an estimated value for this k parameter. And there's a useful website for doing this. I just need to track it down. That we use a lot for estimating this parameter. There was one point where it dropped offline for a bit, and we were kind of bereft, and we had to figure out that we had to kind of reverse engineer the maths. So I do have all of the equations for doing it kicking around in various places, but it's easier to just be able to plug some numbers into a web form. So I am just looking for where this website exists these days. If you just bear with me for 2 minutes, I should be able to track it down in some of our documentation. So I'm getting there. Slowly but surely. Okay. Here we are. That's what it's called, yes. So I'm getting there. So this is a website which was built quite some time ago. It's been around for a long time, and it's got the maths. It actually does have the maths here, which is used to estimate this parameter. It's built by a bioinformatician in the States called Torsten Seaman, who some may have heard of. And the idea here is you plug in some numbers in the top section here, and it gives you an answer for a value of k. This was originally written for estimating this parameter for a genome assembler called Velvet, which is the kind of first assembler which used this idea of Bruijn Graphs for doing assembly. It's quite long in the tooth now, and generally works with only very short reads, but nevertheless, this site continues to be relatively useful for this kind of calculation. So if we remember, we have 1.4 million reads. They are paired end reads. Each read is 208 bases long. And our genome is about 4.7 megabases. And as we put those numbers in the top here, we get some output down the bottom here. So we get a recommended value of k coming out down at the bottom here. So 175, which is quite a large value. The reason for that being such a large value is we have very for the size of the reference genome, we have a large yield, so we have a large number of bases that we've generated in our sequence experiment, which gives us a very large fold coverage, average coverage of our genome. Because of that, because we have this very deep coverage, actually we're not going to need to divide our reads up into very many bits, because the coverage is very deep. So k ends up being quite close to the length of the reads in this particular instance. The other thing that we have control over here is this thing called this fold k-mer coverage value here, which is described down at the bottom here. The kind of there's a relationship between k-mer coverage and standard coverage, but k-mer coverage is how many times a k-mer is seen among the reads, which is different to the actual coverage of the genome itself. You can vary this parameter, so we could make it 30 rather than 20, and it would change this value of k. There isn't really a correct answer for what to put in here, and obviously changing it is going to affect the value of k in various ways. I'm going to use 30 in this instance just because I've got a lot of reads, so I've got a lot of depth, so I would expect my k-mer coverage to be relatively high. I'm going to stick with 30, but you can vary this, and with the assembler that we're going to use, you can use multiple values of k you can use multiple values of k to test different assemblies, essentially, and that means that you don't necessarily have to just take one value here and use that as the value across in your experiment. You can actually pick more than one and use multiple values of k should you so desire. Okay, with that in mind, it's on to the actual assembler then. So the assembler that we're going to use is called SPAdes. So it's developed by a group in St. Petersburg, and the SP in SPAdes stands for St. Petersburg, I believe. But it's a relatively commonly used and modern genome assembler that we quite often recommend as a good kind of utility all-purpose assembler for genome work. So we're going to have a shot at using SPAdes to assemble this data. Now, my disclaimer at this point is that I've not done this before with this data, so it may be that this runs smoothly and goes well, or it may be that I set SPAdes going and we're waiting for the rest of the session for it to finish. So we'll see how we go. But the first thing we need to do is we need to install SPAdes, and for that we're going to use Conda. So we're going to create a new Conda environment. So let's yeah, that's it. So here we go. So let's have a new Conda environment. So Conda create -n for name, and then the name of our Conda environment is going to be assembly. So we're just creating an empty Conda environment with no software installed in it. So that shouldn't take very long, he said naively. Yep, there we go. And then we're going to activate that environment. And then SPAdes should be available in the Bioconda channel, which we have added as a default channel in our installation of Conda. So Conda install SPAdes should hopefully trot off and find the SPAdes assembler for us. So there you go. So in Bioconda there, we're finding a version of SPAdes. So let's install those packages, which should get us SPAdes at the command line. Away we go, slowly but surely. Okay, good. So Conda has finished, and we should now have a SPAdes executable. The executable is actually a Python script, and it's so it's spades.py. That's our SPAdes executable. So that's the thing that we need to run in order to run SPAdes. So if we run it with no arguments, we get some help information. So here we get our usage, so spades.py, then some options, and then define an output directory. In the options, we've got all of these various values that we can define. So let's work our way through that and build a command for doing our installation here. So for doing our assembly. So spades.py. Then we want an output directory. So let's call it. Let's just call it test assembly. That'll do for now. Okay, so some options. What do we need? We need to tell it where our reads live, for one thing. So to do that, you use the argument names. They're very similar to the argument names for bowties, so -1 and -2 for your forward paired end reads and your reverse paired end reads. So let's give it those. So -1 for our forward reads. So SRR 118666736_1, and -2 for our reverse reads, or our second in pair reads. So there you go. That's our second in pair reads. So that's kind of the simplest place to start. So that's just our data. There are lots of other ways of defining the data, and they're all listed here. There are also different modes that you can run SPAdes in. So if you've got high coverage isolate data, for single cell, assembling data from single cell genomes, metagenomes, plasmids, RNA sequencing, there's all sorts of different modes that you can run SPAdes in. We're just going to run it in the default mode, so we're not going to define anything for that. Then we need to tell it, so we don't need any of the rest of these things because we're not giving it any trusted contigs or anything like that, which you can do if you want to give it a reference genome to assemble against. We could be careful, but that's going to slow it down, and I want to try and get through the assembly in the next 25 minutes, so I don't necessarily want to. Although I would, in lots of ways, running it with careful switched on would probably be sensible. I'm not going to do that right now. You could if you're doing this at home. Okay, number of threads. Default 16. I'm not not sure where it's getting that from. I'm going to have to run it with one thread because my virtual machine only has one thread. RAM limit for SPAdes in gigabases. Default is 250. My machine's only got 8 gigabases. So let's give it a memory limit. All of these things are probably going to slow it down. Right, so K, list of Kmer sizes. So you can give it a list of different Kmer sizes. It says must be odd and less than 128, which is where we're going to run into slight problems here, because our recommended value for K is 159, which is larger than 128. So that's going to make our life slightly problematic. So we can tinker with our K-mer coverage here. We've got quite deep sequencing, so maybe we could play with this until K drops below 128, and then we could use that number. We could also use a range of values of K below 128 and see what comes out best. In the interest of time, I will estimate 50-fold K-mer coverage, which is slightly a fudge factor in this case, and we'll use a K of 125. I'm not necessarily saying that that is the best value for K to use, but we don't really have time to go into too much detail at this point. The other thing that I would like to do is just set this this last thing, which is the coverage cutoff. So cov cutoff, which I'm going to set to auto. So it's going to auto detect it in my data. This is a value by which, if you have bits of your genome that are covered below a certain threshold, then they won't be considered in the final assembly. And that's important for kind of getting rid of fragments that are assembled out of just a couple of reads and things like that. So we're just going to set that to auto for this particular assembly, just so SPAdes can figure it out for itself. Now, I suspect this assembly may not run terribly quickly, but we're going to give it a shot, and we're going to see how it goes over the next 20 minutes or so. Hope that it completes. And while it runs, I'm going to talk about how we assess assemblies once we've got them completed. Okay, so the SPAdes is now running and is going to run for, I suspect, a little while. I'm hoping it will be quick enough that we can have a look at the results before the end, but it may well not be. So let me give you an idea of what an assembly might look like, and how we can assess the results of a genome assembly experiment. I'm just looking for an example of one which I have done before, so that I can show you what the results of SPAdes looks like without us necessarily having to wait for SPAdes to finish running. So no, that's not one. Getting there, don't worry. Here we go. Maybe. Yes, here we go. Right, so let me show you this on one of my machines. Here we go. So this is an assembly exercise that we used to do in one of our courses, and we've run SPAdes over this data here with 3 different values of K. So we've run with a value of K of 83, of 95, and 107. So this is a different data set, different bacteria. And what you get from SPAdes is quite a lot of output. So you get an assembly for each of your values of K that has been that you've chosen. So that's what those directories are. So we've got a K83 directory, which contains our assembly for the value of K of 83. and we've got the same for 95 and 107. And then SPAdes estimates which of those assemblies is the best and comes up with this final output, which is essentially your assembly, and that is in this file which is called contigs.fasta. So contigs are what your regions of contiguous sequence that you're attempting. That's what you're attempting to build when you do your assembly. So this contigs.fasta file is the primary output of SPAdes. So in this instance, we're looking at the assembly of a different genome in a different experiment. So here we have a contig, and the naming that SPAdes applies to contigs is fairly standard. So each contig is called a node, and they're numbered. So here we've got node 1. It tells us how long it is. So this particular node is 443,000 bases long. So 443 KB. It gives us the coverage of that node. So the average coverage of this node in this experiment is just a bit shy of 19. So every base in this contig is covered on average by 19 reads. This is the first contig in this genome assembly, and we can scan through this to find the next contig. So and Dan is asking, is the scaffolds.fasta not considered the assembly? So in a de novo SPAdes assembly like this, where you have you've not given it any trusted or untrusted contigs from a previous assembly or from a reference genome, the contigs and the scaffolds will be essentially the same, because SPAdes has nothing to provide the scaffold with, so it can't decide on the placement or the distance between the contigs, and therefore generate scaffolds, because it has nothing to base that on. So as far as I'm aware, there will be little or no difference between the contigs and the scaffolds in a standard de novo reference-free SPAdes assembly. 400,000 is more bases than I thought it would be. I'm paging up through a lot of this file. Let's use less's search function. So I've typed / and then the greater than character, and that jumps me to the second contig. So here's the second contig, so node 2. Its length is again 400 KB or so, 412 KB in this case. And its coverage is similar to the first one as well. So as the coverage for the first one was about 18, the coverage for this one is about 20.5, 20.6. So coverage of those first two contigs is very similar. Their length is very similar, but these are two different bits of the genome that we've assembled here. And then node 3, again, relatively long, 250 KB, and similar coverage, around about 20 for the coverage here. And to a certain extent, this is what you're looking for in your assembly, is that the contigs that you build when you're doing an assembly are relatively long, so you want them to be you want your average contig to be longer than your average gene, really, in an ideal situation. So you're looking for certainly thousands, preferably tens or even hundreds of thousands of bases. You want the coverage of those contigs to be pretty even. So in an ideal scenario, each of these contigs would be covered to around about the same average depth. And if you had contigs which vary wildly in their coverage, then that would be reason to suspect the quality of your assembly. So you can see that for all of these big contigs in this assembly, the coverage is around about 20, which is good to see. So you've got some nice long contigs covered in a fairly even way. So this is looking like a relatively decent assembly for this particular genome. So we've got quite a few contigs here, but they're all of these are pretty decent length. They're all up in the hundreds of kb, and they're all covered at around about 20-fold. So this is looking okay as far as a genome assembly goes in this particular instance. So the other thing that you can do with an assembly is you can try to assess the quality of it both on its own merits. So here we're doing this by eye, by checking out these contigs, but you can also do this computationally. So there are metrics that you can apply to look at a genome assembly, which will tell you about the quality of that assembly. So one of those is the so-called N50 value. I'll talk about what N50 means in a minute, but we can generate it for this. I've got on this machine, I have a tool called GNX, which is installed, which you can use for calculating the N50 of your assembly. I'm going to exclude any contigs smaller than 100 bases. I want to calculate the N50, so NX50, and I'm going to give it my contigs file to calculate it on. Okay, so this gives me some summaries about this particular assembly. So it's telling me how many sequences there are in total in my assembly. So here we've got 64 sequences in our assembly that are longer than 100 bases, that is, so 64 contigs. If we add up all of those contigs together, we get an assembly of 2.8 megabases, so 2.8 million reads. 2.8 million bases. Our shortest contig is 108 bases. Our longest contig, which we've already seen, is 443,000 bases. And the N50 is 150,907. And it says there are 5 sequences encompassed in that N50. So just to explain what this N50 metric is, and I'm going to need my whiteboard back for this, so let's hope I can get this working again. Okay, just hide things while we're rearranging my screens a bit. Okay. Let's refresh that and refresh that. And check that it's working. Yes, good. Okay, right, so N50. So we have produced an assembly that has a range of contigs of different sizes. So let's come up with a kind of superficial example for this, sort of back of an envelope. So here we've got a small number of contigs of different sizes. That will do. Okay, so there we go. So we've got a small number of contigs of different sizes, and we want to know what the N50 of this assembly is. So there's a few things that we need to do in order to be able to calculate that. So the first thing that we need to do is we need to figure out the total size of the assembly. Okay, so we need to add all of these contigs up. So we have 6, 8, 14, 15, 16, 21, and then 2, 7, 10, 11, 12, 13, 15. Okay, 1,516. That's how big our total assembly is. And then the next thing that we have to do is we have to order these contigs from largest to smallest. Okay, so the largest is 500. The next is 360. Then 212. It's not ignoring my hand resting on the tablet, this particular whiteboard, which is interesting. So you'll see all sorts of things appearing because I can't keep my hand off the screen. So that was 212. Then what's next? 150. That one. Then 124. No, I've crossed out the wrong one, but you know what I'm doing. 120, 100, and then 70. Okay, so those are my contigs in order, and now we want to add these up. We want to keep a running total, and we want to take a running total until our running total is bigger than 50% of our total assembly. So our total assembly is 1,516 bases. So 50% of that is 758. Yeah. So when our running total gets bigger than 758, that's where we want to stop. So running total of 1 is 500. The running total of 2 is 860. We're already bigger than 568, so that's where we stop. So that went very quickly, but nevertheless, what that tells me is the N50 of this assembly is 360. Okay, that is the size of the contig where my running total of adding up the sizes of these contigs in size order went over this number. It went over 758. Okay, so once I got to more than 758, the size of the contig that I just added on to my running total, that is my N50 contig, and the size of that contig is my N50. Okay, so in my particular hypothetical example here, the N50 is 360, and that will encompass 2 sequences. In the example which I showed you in my terminal here, the N50 is 150,907, and that encompasses 5 sequences. So that means the 5th sequence is the point at which your running total crossed that 50% threshold, and that therefore is the N50 for that particular assembly. There are other analogous values to the N50. So there's the basically any number you can think of. You can have the N25, where the running total gets bigger than 25% of the total assembly, or the N75, where the running total gets bigger than 75% of the total assembly, and so on. This tool GNX can calculate those numbers as well, either the N25 and the N50. These are also referred to as the NA value, so the NAX value, so the NA50, and that's because it's the N50 of your assembly. You can also, if you've got a reference genome, calculate the NG values, and that is the same thing, but instead of using the total size of your assembly as your reference point, you use the size of a reference genome as your reference point. So you're looking for the point at which your running total exceeds 50% of your reference genome size. Now, in an ideal world, your reference genome and your assembly should be fairly similar sizes. That would at least be the hope. There is another tool, so I've shown you GNX here, but there is another tool which is developed by the same people as SPAdes, and that's a tool called Quast, Quast, which produces visual output for these kind of metrics as well, and is a really nice tool for assessing the quality of assemblies. I have, I think, run Quast over these samples. Yes, I have. So let me just see if I can get that. Let me see if I can get that output and show you what that looks like, just as a last thing to do before I finish. Just to show you, SPAdes is still running and has been, so it had been running for 7 minutes when it got to this current point where it's building a clustering Hamming graph. So it's done quite a lot of stuff up until that point, but it's been doing that for quite some time now, so I don't think it's necessarily going to finish anytime soon, which is a little bit of a shame. But let me show you the Quast output for this other genome. So I'll just very quickly try and get hold of that to show you what it looks like. I think this was produced using quite an old version of Quast, so it may well be that that the output is different to this now, or maybe richer than this now. But nevertheless, I think it's worth showing you what I've got here, just because I do at least have it. Whereas obviously we're not going to get to that point with the assembly that we're generating today. So I'm just copying that across to my current machine, so that I can show you it. Let me just do this, because otherwise my screen's going to jump around, I think. Right, okay. Going as fast as I can, I promise. Here we go. Then you want to go there. Then there, that's it. And there. And then, right, it's just taking a short amount of time to download. Some of the files that Quast generates are quite big, so now I'm getting stuff out of the university to my home machine. It takes a little bit longer than the internal transfer took between machines at the university. Hopefully it's not going to take a huge amount of time, and I can just show you this as our very last thing today. While this is downloading, I just want to talk about next week briefly. So next week, two things. One, it's the first week of the school summer holidays here, as I'd already said, and which means that I think that lockdown learning will be happening slightly later than the usual 4 p.m. BST, because I'm going to have children to coordinate in a way that I haven't had while they've been homeschooling. So I think that we're going to do this slightly later next week, or possibly on a different day. But I will stay tuned for updates on that, and I will make sure that I announce it on the Slack and on Twitter, as well as maybe some other places. Also, I'm planning on doing essentially an Ask Me Anything, so in the vein of the Reddit Ask Me Anything kind of thing. So what I'd like to do is gather some questions from lockdown learners before the live stream next week. Things around bioinformatics which you might want answers to, which maybe I haven't covered in the sessions, or maybe clarifications of things which I have covered. Also, if anybody is, and I had a suggestion about this on Twitter yesterday when I kind of suggested that I might do this. If anybody's interested in just my experience of the live streaming and of lockdown learning generally and how it's worked, then I can also answer those kind of questions as well. I'm kind of throwing myself open to all sorts of things, but that's why I'd like for people to send me questions in advance if possible. So send me direct messages on Twitter, private messages on Slack, emails, whatever you want. If there's anything that you want to ask me, if you have any burning questions, then please feel free to send them through. That session will only be as rich as the questions that I receive, essentially. So that's my plan for next week. And as I said, it may not be at the normal time, and so I need to figure out what the schedules are for next week and try and figure out when the best time is going to be to do that. But that's the plan for next week. So here we go. We've got some results from Quast here. So this is a Quast report. You can see I generated it quite some time ago. This will have been on a course which the BSU ran here in Newcastle. I gave Quast a reference genome. So we've got a reference genome with a size and a GC content. And then we've got our assemblies here, which have been generated by Quast. That's a good question. Can you provide a new link to Slack as the old one has expired? Yes, I absolutely can. I will. I'll figure out somewhere to post that. Probably I can put it in the description of this video. Okay. Yes, here is the reports that we get. So we get the total length of the assemblies, the length of the largest contig, the number of contigs, and we get some interesting things about misassemblies. That's compared to the reference genome. We also get, you can see here, reports about the various, the size of the contigs. You've got the running total here as we add the contigs up. So as we add the contigs up, we get the kind of running totals. And if we get the extended report, so there we go. We've got you see the NG50. So that's the size of the contig where you cross 50% versus the reference genome, as opposed to the NA50, which is the same for the assembly. And you can see that those numbers differ. It's just this is quite an interesting and useful report on your assemblies that you've built. Here, obviously, we've got multiple assemblies, so we've got our different lengths of K, which give different assemblies. So for those all have different statistics, and you can use the Quast report here for trying to figure out try and figure out which of those assemblies is the best assembly either compared to a reference genome or just on its own merits. So Quast is quite a nice little tool, and there is a Conda recipe for Quast, so you can install Quast using Conda. And then run it versus our SPAdes assembly once SPAdes has completed. And obviously SPAdes is not going to complete within the time frame of today because I'm going to stop. So hopefully that's been interesting and useful to people. Something a little bit different to some of the things that we've been doing both recently and kind of overall as well. Genome assembly is a little bit different to the other things that we've been doing, which have been kind of more alignment focused. So, as I said, next week, ask me anything, get some questions in. I'll set up a Slack channel. I'll redistribute an invite link to Slack to make sure people can get on there. There's other venues to get hold of me as well. Obviously, Twitter and various others. I'm available. So yeah, get me your questions, and if I get some good ones, hopefully we'll have a really good session next week. So I'll see you then. So everybody keep safe, stay well. See you next week.